Hello, and thank you again for inviting me to present today. My name is Matteo Rizzi, and I'm a group leader at the UCL Institute of Ophthalmology and Moorfield Eye Hospital. So my research group works on developing gene therapy. We try and develop therapies for various forms of blindness. And today I'm going to be talking about a form of macular degeneration, which is Stargardt disease. The approach that we have in the lab is very much to first understand how patient vision is affected, and only then, as a second step, we design our gene therapy. And the work I'm going to present today is very much split in that way. So the first half is to try and understand how a condition like Stargardt disease affects vision, and then how we can design therapies for it. So in this second slide, I put a picture of some of the members of my team whose work I'm going to present today. And people are the most important ingredient, of course, of good research. So without them, this work that I'm going to present would have not happened. And then when people join the lab, I usually show them a graph, which I'm showing here on the slide, which tells us how much gene therapy is an emerging field, and there is a sudden increase in the number of clinical trials for gene therapy over the last few years. And what we'd like to do with the lab is to make this grow even further, together with other labs across the world, of course. So as I said at the beginning, our work is split between Moorfield Eye Hospital and the Institute. And there is actually a physical bridge that connects the two buildings, which is very important for my research group because we go back and forth a lot between the hospital, where we try and talk to patients and test their vision and understand how it's affected by their condition. And then we come back to the Institute to develop what we call models of the condition, to try and understand it, and also to try and test our therapies. So Stargardt disease is the most common form of inherited blindness. It's still a rare condition. 1 in 8,000 people are affected, but it's the most common form. And unfortunately, it hits the central vision in patients. What that means is that it affects the vision with the highest resolution that we have. Our eye is not uniform in how good it is at seeing details, and the central part of the eye, as you may know, is the one dedicated to vision with high resolution. And unfortunately, that's the part that is affected in Stargardt disease. If we imagine, it's obviously difficult to put ourselves in the shoes of someone with macular degeneration or Stargardt disease, but if we try and imagine how their vision is based on what they tell us, there is a blind spot in the middle in this high resolution region, which is obviously very impairing for their daily life. Now, a lot of great work has been done with Stargardt disease, particularly from geneticists who have pinpointed that there is a specific gene called ABCA4 that is responsible for Stargardt disease. So it's a very large gene, and that means that there is a lot of possible variants in this gene. All of us have variants in our genes, but unfortunately, some of these variants in ABCA4 are associated with Stargardt disease. They lead to Stargardt disease. I think it's an interesting example of something that we have understood very, very well from the point of view of genetics, but from the point of view of function, we're still not 100% sure what the ABCA4 gene does in the retina normally. There are some very good hypotheses. There is a lot of interesting data that suggests what probably it does, but we're not quite sure yet, and a lot of work, including in my lab, is ongoing to understand that. This is not necessarily a problem for developing a therapy, because once we know which gene is affected, we can go and design a form of gene therapy. So even if the field is catching up, we're trying to understand exactly, precisely what this gene does in our body. In the meantime, we can develop gene therapy approaches. So one thing that we have discovered in the lab, and I wanted to share with you, is that if a patient loses vision centrally, of course, that part of their field of view will be a blind spot. There will be no vision there. But we're finding that actually even the surrounding areas of the retina, parts that are not directly affected, are not directly degenerating, or not yet at least, they are also affected. The kind of metaphor that you may want to think of and that we usually use is that of the pebble in a pond. So if you throw a pebble in a pond, you will see these concentric circles that expand out in the water. And so similarly, the effect that there is in a certain spot at the back of your eye spreads through specialized circuits that we have in our eye. It spreads laterally in concentric circles, very much like a pebble in a pond or on the surface of water. And the result of this, to cut a long story short, is that the remaining vision around this area of macular degeneration, around the area that Stargardt disease primarily affects, this area is also affected and vision is not as good as it could be otherwise. So like I said, I'm showing on this slide a schematic showing a simplified version with degeneration in the center of the eye. But there is an area around it where vision could be better. And so while patients with remaining vision have a blurred vision, worsened vision in these areas, it could actually be quite a lot better. And if you think of the classic doctor's eye chart, I am talking about a difference that could be up to 5 lines on this classic doctor chart. So you'll be able to read, for example, much better if there wasn't this problem, this lateral problem in the remaining retina. So some of the work that we're doing, as I said at the beginning, is to try and understand the mechanisms behind this. How is it possible that degeneration in one spot of the eye also affects remaining vision around it in the healthier part of the eye? And the work that I want to talk about is the work by Hasina Abbar. Hasina is a scientist and also is someone that several years ago was diagnosed with Stargardt disease. And so this gives Hasina a very unique perspective because she can, of course, understand personally what it means to have Stargardt disease and how her vision is affected, but also to design the experiments and perform the science to try and dissect this and understand it better. So when we first started speaking with Hasina and discussing work that we could do together, Hasina reported to us that this central effect of Stargardt disease, the effect that the condition has on her eyes, was causing 2 symptoms that seem to be underappreciated by the clinicians she has spoken with. One of them is the experience of light flashes. If any of you have the same condition, you might have also experienced it, or other conditions are also associated with this. Essentially, it means that the patient perceives flashes, even if in reality there is no light flashing in that moment in the world. So there is no stimulus, there is no blinking lights, and yet the patient has an illusion that the person thinks that they are seeing a light flash. This could be, from what Hasina told us, it could be of different shapes and different locations. So it could be a single big flash, it could be lots of little sparkling lights. It could be apparently also sometimes shaped a bit like a spiral or like the letter C. But in general, it seemed to be a feature, a prominent feature, and quite a scary one, apparently, because when something comes out of nowhere, of course, it can be scary for us. The other symptom that Hasina mentioned to us is an extreme light sensitivity, which some people call photophobia, but I think that's a bit of a misnomer because it makes us think of other type of phobias, and it's not quite the same. From the description that we got from Hasina and then from other patients, this is more the idea that you very easily find light too bright if you have this condition. If you walk outdoors, for example, things look brighter than they would to a person with an unaffected eye. And so as a result, for example, people end up wearing sunglasses all the time, and if they don't, they find this quite annoying, and they might develop a migraine or a headache over the course of a day. So we thought this was interesting because for details that I'm not going to go into today, we thought this would link well with the pebble in the water mechanism that I described before for you. And so the first thing we did with Hasina was to try and think, well, could this be just Hasina's experience, or could it be actually that many patients have this? So she structured a questionnaire, which was then delivered to several patients. We did a first cohort, and now we're going to do an even bigger cohort. And the first cohort was delivered to patients via the charity Stargardt Connected and through Moorfield Eye Hospital. And what Hasina found was very interesting, because it turns out actually that the vast majority of patients with Stargardt disease that we sent the questionnaire to do have the experience of light flashes. About 90% of them, in fact, have experienced this. And even this excessive light sensitivity and light aversion is in the majority, in about 2/3 of the patients. So I'm going to give you a few more details about the work Hasina did. One thing is that at the end of the questionnaire, she left a box where people could leave a comment. And it was very interesting because some of the comments are very interesting because a couple of the patients, for example, reported how underappreciated this was, as I said. And so there, one of the patients said, I'm glad this is being looked into because I feel like it's connected to my Stargardt disease condition, but I've been told that it's not. So Hasina also looked more in detail about the quality of these light flashes and these symptoms. And so just to give you an idea of some of her work, we asked, for example, patients what triggers, if anything, triggers these flashes. And it seemed to be that the most common response was that a change in luminance was a trigger for this. So if people switched on a light in a dark room, or they went out from a dark room into light, or vice versa, changed from back from light to dark, this seemed to cause the light flashes more than other issues. Stress and fatigue, of course, were another trigger, and so on. And when we asked them instead the typical duration of these light flashes, we actually found a broad spectrum. So some people said that this is only for seconds or even just 1 second. Other people instead said that unfortunately this is constant and is there all the time, or at least there for many hours. So Hasina is going to start soon this PhD project. This was the preliminary data that she collected, but already now we are seeing some interested connections, some links with the stage of the condition. For example, she found that if many years have passed since the person was diagnosed with Stargardt disease, this symptom of light flashes increases significantly. And another thing that we can do, because as I said, we work at Moorfield Eye Hospital, is to try and link what the patient tells us with then some measures in the hospital. This is very important to try and find the mechanism behind this. So when we did the questionnaire, we asked participants where in their field of view, like if they imagine looking at a picture, where in that picture they would have the flashes. And very interestingly, there was a hot spot in the center of vision, which is very consistent with the idea that this is not a generalized problem in the eye of patients, at least in the majority of them, but it's something really that is linked with this degeneration occurring in the center. Again, you can go back to the metaphor of the pebble in the water. So when you throw the pebble, the area of water that is most affected is the one right around it. And in the hospital, we're going to do some measures that allows us to present stimuli in very specific spots on the eye of patients. And so we will be able to tell how different parts are affected by these light flashes. Now, I want to also briefly touch on something that I think is a very exciting aspect of research and of our research in particular. Because after we have conversations with patients, after we do a questionnaire with the patients, it's very important that we turn these words into numbers, that we turn these words into a proposal, a suggestion of what the mechanism might be behind this. This is not only to have a hold and understanding on the condition, but also because if we understand the mechanism, then we will be able to design a therapy for it. And so one thing that many people do, and we've done as well for this, is to create what we call a computer model, which essentially means ask a computer to try and replicate, to try and mimic the way the images appear to patients based on what patients tell us. So we give the computer just enough details, and that it is important to know whether those details are sufficient to explain and lead to the same conclusion. This is work we did with our colleague Martha Robinson, and actually what we found was that the computer had a very similar opinion to what we hear from the patients. So by putting in the right kind of details that we think can explain this phenomenon, we were indeed able to show that the computer creates images that are very bright, essentially. So it can mimic this experience of having excessive stimulation and light aversion. Now, as I said, the second part of the work that we do is to develop gene therapy. And so based on work such as this, like what I've shown you from Hasina, we try and design gene therapy. So in the second part of my talk, I want to focus instead on the gene therapy approaches that we try and develop based on these studies and this understanding that we gain thanks to the conversation with the patients. So one type of gene therapy that we're doing is to try and improve vision in the remaining vision in Stargardt disease. And to do this, we are targeting the cells. I'm showing here a picture of these horizontal cells that they're called. They are cells that spread horizontally in the back of our eye. And without much creativity, we're called indeed horizontal cells. And these are cells that we discovered in our lab to be very important for this pebble in the water mechanism. So these are cells that are very important to make sure the remaining vision in the eye of patients is as good as it can be. And so what we're doing is design a gene therapy approach to target these cells, to deliver a specific molecule to these cells. And what we think is that we'll be able to make vision in these patients better. But we're still at the preclinical stage, so we haven't tested this yet in a clinical trial with patients. Now, I guess you could also raise some criticism, because trying to improve remaining vision in patients with Stargardt disease might be a great goal, but at the same time, it would be better if the disease hadn't developed in the first place, or if we had been able to stop it earlier on. Why wait for the degeneration to occur and then make vision better, right? So ideally, if we could develop a gene therapy that acts at the earliest possible time, that would be, of course, much better. And so this is what we're doing in a parallel project, and I will use the rest of my time to talk about that. So what we use for gene therapy, and in the case of Stargardt disease as well, is what we call vectors. So vectors are little carrier molecules that essentially take your gene, in this case is the ABCA4 gene that I've shown you at the beginning. They take the healthy copy of the gene and they put it back into the cells in your eye. There are many different types of vectors, and some of them are large, some of them are small, some of them have a short-lasting effect, and some of them have a long-lasting effect. Of course, in terms of duration of the effect, as you can imagine, the longer the better. Ideally, we just want to do a single injection in the eye of patients, and then for all their life, they will have this healthy copy of the gene, and they will not have a Stargardt disease anymore, or it will be very much slowed down. So the vectors that we're thinking of using, or that ideally we would use, are the ones that have a very long-lasting effect. At the same time, though, the size matters, and it matters in particular for Stargardt disease, because the ABCA4 gene is a big gene, is a very large gene, and that means that we can literally physically cannot squeeze it into small particles, into small vectors. So that means that for Stargardt disease, we need a large vector to carry this large gene. There is such a vector, and it's called lentivirus, and it's based on lentivirus. We call it lentiviral vector. And I'm making it sound, I guess, like everything is perfect, and the problem is sorted. We have a vector that is suitably large and that lasts a very long time, probably forever, in the eye of the patient, so it sounds like a dream. The problem is that this vector is really not very good at entering cells of the eye. It's very good in other parts of the body. It has been used already. It's constantly used for other cells, such as cells in our blood. But it's really not very good for cells in the eye. And so what we're trying to do in the lab is to try and push the ability of this vector. We want to make this vector, the lentiviral vector, as good as any other vector, maybe even better at entering the cells in the eye. Because then it will mean that we have a vector that likes the cells in the eye, that is big enough to have the gene that is needed for Stargardt disease, and also that lasts a long time. So that would be an ideal scenario. So how do we find, how do we make this vector like the cells in the eye? And I think I want to make a general point, which I think this is a time in science where we humans, we scientists, are realizing how small we are compared to nature, for example. So now we have these big libraries of molecules that have been discovered in all sorts of realms, in all sorts of locations on the planet. And we're really finding that evolution has created a huge number of variants of all types of molecules. So what that means is that instead of us trying to be overly clever in the lab and think that in a few months we can come up with the same that evolution did in millions of years, we can actually just go and screen some of these available molecules from this library, let's say, that evolution has created and nature has created, and pick from there the best molecule to do the job for us. The second thing that we can do today, and this is a very recent development, is that, as you know, artificial intelligence is becoming a very powerful tool. And artificial intelligence, or AI, is very good at creating huge libraries of information, and then also at looking at scanning these huge libraries. So in a way, we can also have this second huge library that we can ask help from to try and understand which of the molecule designs, which of the conformations of molecules will be ideal for our task. And so, for example, in our group, we use both these screenings, these looks that we have at the huge library of evolution, and at the same time also looking at huge libraries created by artificial intelligence to find the ideal molecules that are going to help us improve our gene therapy vectors. And what I'm going to very briefly mention today is a case where we looked at the first library, the library of evolution, to try and help with Stargardt disease. So as I said, there is many millions or billions of molecules that we could be looking for. But what we found, and I'm cutting a very long story short, of course, but what we found is that there are at least 2 molecules that seem to help these vectors to like finally the cells in the eye, to finally enter the cells in the eye. And so this means, in very simple terms, what this means is that we are literally mixing in a vial this special molecule together with our vector that carries the healthy copy of the ABCA4 gene. And the mix of these 2 things makes the vector and the ABCA4 gene go very efficiently into the cells at the back of our eye. So this is great because finally we can do what I was saying just a few minutes ago, which is we can make this vector like the cells and deliver the gene therapy to them. And in this last slide, I am showing some of the data that we are collecting in the lab. So in white, I'm showing the light-sensitive cells at the back of the eye, in the retina. And in green, it's a special labeling procedure, a special way to have a signal from the gene ABCA4 and from the protein ABCA4. And so we are able to see that in an unaffected eye, ABCA4 is there. In an eye without Stargardt disease, this band is gone. ABCA4 is not there anymore. And then we're very excited that with our approach of mixing, like I said, this molecule that evolution has created naturally, together with our vector, we can finally put back a similar amount of ABCA4 gene with gene therapy in the back of the eye. This is also still preclinical work. Again, we are not at the clinical trial stage yet, but we're hoping in the next few years to go in that direction. And the last slide I want to leave you with is a slide with my email addresses. And I want to just say that we're always very happy for people to visit our lab. So please get in touch if you're interested. I think, as I've shown you in the first half of the talk, for us, it's also very important. It's not just to tell people what we're doing, not just to be open, but also because it's really very important for us to start from that, to start from understanding how patients see the world, to try and then go on and design our gene therapies. So thanks for your attention, and I'll be happy to take any question you may have.